So good morning, everyone. I'd like to thank the organizers for inviting me to talk about schistosomiasis. Maybe a few words about the group. So we work in the global health unit from Merck Serono, which belongs to Merck. We also a very newly formed global health group, maybe 1 year old. And I, as Cameron just said, I just joined like 3 months ago the group to lead drug discovery activities. Is this one? Yeah.
This one, yeah. That's it. There we go.
So I'm going to talk to you about schistosomiasis drug discovery. Where next? But before that, maybe let's talk about some facts about schistosomiasis. So it's a disease that's caused by a flatworm of the genus Schistosoma. Schistosomiasis, also known as bilharzia, is a chronic disease, and it's due to host immune response to the eggs laid by the adult female worm. We can distinguish two types of schistosomiasis. One would be the intestinal one, with S. manzoni and S. japonicum being the main species in subtropical and tropical areas, with some complications such as liver and periportal fibrosis, hypertension, hepatomegaly, intestinal fibrosis, diarrhea, blood and stool. And the other one, which is urogenital schistosomiasis, caused by S. hematobium, mainly in Africa and Middle East, causing urinary tract fibrosis, bladder cancer, and renal failure. More than 200 million people are affected. 90% are in tropical and subtropical areas of sub-Saharan Africa. 50% are children. The burden is ranging between 12,000 and 200,000 deaths a year, according to some references. Importantly, in terms of socio-economic impact, schisto ranks second after malaria, according to WHO. And as of today, we only have 2 approved treatments by WHO, which are both monotherapies, one being oxamniquin, dating from 1967, actually only efficacious in South America against S. manzoni, therefore used only in South America, and mainly South America, and praziquantel from 1975, which is used for MDAs, although the coverage is only 14.4%. So what is actually Merck doing in the schisto world? So we've signed, like many others, the London Declarations on Neglected Tropical Diseases. As you may know, we've been active since 2007 in a donation program of praziquantel, and we aim to reach about 250 million tablets a year in more than 20 African countries. We also work in the fight against fakes with the Global Pharma Health Fund Mini Lab, with a kind of a small suitcase that contains non-sophisticated low-cost screening methods based on chemical testing to detect the fakes. In a consortium, we also work together to deliver a pediatric formulation of praziquantel and L-praziquantel, thought to be the active form of praziquantel. And finally, we want to work on some drug discovery efforts for schisto within our global health units, by building a mini portfolio to fill the therapeutic gaps and actually reach the elimination agenda. Talking about elimination agenda, you will soon hear about the launch of the Global Schistosomiasis Alliance end of next month, if everything works well, with the vision of eliminating schisto as a public health issue, according to the World Health Assembly Resolution 65.21. The mission, again, is here to strengthen the impact and efficiency of the global response, again, working in country-led type of efforts with integrated and innovative solutions. And the approach, again, very much integrated through partnership platforms with the public and the private sector, again, across the globe. Merck is one of the founding members of the Alliance, and just to note that there is also a working group that will focus on R&D efforts for schisto. So, in terms of pipeline, I mean, according to literature at least, it's virtually empty. Several reasons behind that. Schisto has probably less impact than other diseases because it's silent at start, it's chronic, and giving the wrong perception that it's a less dramatic disease. Also, we have, luckily, praziquantel, which is readily available, safe, efficacious, giving the perception that there is actually no need for a new drug. However, when looking at praziquantel, the mode of action, even 40 years after use, is not very clear. The PK-PD relationship not completely understood as well. Praziquantel is only active on the adult worm, so meeting the elimination agenda seems quite difficult just by eating the adult worm. After 40 years, no major signs of resistance, although if one would do a true mass drug administration, you would definitely increase the drug pressure, and therefore there is a likelihood of emergence of resistance. So many reasons for an empty pipeline. So how could you address that? First, by kind of drafting some drug target profile. So if you look at the right-hand side of the slide, you see the host life cycle. After skin infection, the cerca actually maturates into the schistosomula, which then becomes a juvenile worm before becoming the adult worm. They mate, they produce eggs in the mesenteric venules, and then shedding, partially shedding actually the eggs, which are ultimately used for diagnostics. So ideally we would look for a compound or compounds that have a mode of action differentiated from praziquantel, something that is active against all types of schistosome infections, so not like oxamniquine, for example. Ideally, we would be active on the adult, but also on another life cycle stage, for example, on the juvenile worms, to provide kind of a prophylactic potential to the drug. Also active on the eggs to prevent the pathology or eventually block transmission. Finally, also, praziquantel has a very short half-life, so ideally we would look for something that has a prophylactic side of it, which with a reasonable half-life, or let's say a time above MIC. And finally, to limit the resistance, we would like to have something in combination. So, when zooming on the drug discovery part, if you look on the right-hand side, I mean, the parasite actually evolves in very different, let's say, stages, which are located in different organs and are actually asynchronous. So that makes it quite complicated. So first of all, there's, I think, quite a lot of work to understand really what drives efficacy. What is an active compound? Again, we talk a lot about micromolar type of activity, although praziquantel has submicromolar activity. Also, the in vitro, in vivo correlation is not very clear. The immune system plays a huge role there in the clearance of the parasites. So how can we encompass that in the screening, for example? How would that impact the murine challenge model? Also, in terms of characterizing the compounds, we would like to reflect a phenotypic effect, typically. Also, the rate of kill could be an interesting aspect of characterization of the drug to factor in the PK side. And again, in terms of DMPK consideration, given the fact that it's an asynchronous disease, having something that is long enough and that can target the desired organs. So we would suggest, for example, to work on a target-based approach. The genome sequence of schisto RNA could be used as a tool to document the phenotype. There are already quite a number of putative targets disclosed in the literature. I mean, that's a non-exhaustive list, but we could, for example, hit the motility, tegument alteration, look for the food metabolism, detoxification, respiratory chain, so more in the inner side of the parasites, looking at fertility and production, look at immune evasion, for example, and finally apoptosis. But the essential part is really to validate the targets in a phenotypic assay, of course, with, I think, importantly, in a host mimicking environment. So, that was quite fast, just to conclude. We think that there is a need for additional therapeutic tools for schistosomiasis. We have, luckily, praziquantel, but it's probably not enough to eliminate schistosomiasis on its own, hence the risk of resistance on a mass drug administration. Finally, so we think that there is a need for building and sustaining a mini portfolio of compounds that target the right drug profiles to meet the elimination agenda. And Merck itself is engaging itself very much into the elimination part. We first work on praziquantel pediatric formulation. We think that could be helpful for the next generation of drugs. We're initiating target-based projects. We still work on phenotypic hits from the Merck Mini library, which is a freely available library of late-stage compounds for people to screen. So if you're interested, just come and see me. And also, we're happy. I mean, Wes will talk about it later. Wes and Boris will talk about it later on. It's on the pathogen box, but we think, involving ourselves in pursuing some of the compounds that have anti-schisto activity. And finally, we are, of course, very much open for partnering in drug discovery projects. And I thank you for your attention.